introduce myself as the second speaker. If you can have the slides. Okay, I'm Janos Szebeni from Hungary, working at Semmelweis University, the Nanomedicine Research and Education Center. And I am supposed to give you a quite general overlook on toxicity and safety prediction in nanomedicine. And I am fortunate to refer you to the morning session where we had Raj Bava talking about the issues in nanomedicines, and he quoted toxicity as a second major issue. And why is it an issue? So, reminding you that this is an issue, toxicology, because nanoscience and nanotechnology apparently is moving too fast to enable us to really assess the toxicity, long-term toxicity of materials. So what can we do in this regard? It is difficult to slow down our progress. The solution is to speed up nanotoxicology assessment. And then there is another issue. Can regulations truly tame the vastness encompassed by nano? And I believe he wished to tell with this that there's so much variety in the different nanoparticles. Almost all need individual assessment for potential toxicity, which is just too much for any institute or any organization to take up. And then another issue which is unanswered: does nano pose serious environmental health safety issues? And this is another thing which we cannot know, cannot predict for most nanoparticles because of the lack of time, lack of experience. And it is well known that nanoparticles have unique properties which enable them for unique use in therapy, but they also lend them unique toxicities. And one of such unique factors, physicochemical factors, is the high surface-to-volume ratio as compared to the bulk counterparts. And I'll show some examples for toxicity related to this. And he referred to complement activation, which you may or may not know is our theme, our focus. And I'd like to talk about it some more. And the toxicity of many nanoscale material is not fully apparent, which is also part of the problem. We just don't know what potential toxicities these nanoparticles carry because they have not been studied enough. So pre-market testing of nanomedicines will not detect all adverse reactions, and it is critical that long-term safety testing be conducted. That was Dr. Bava's conclusion, and this is, I believe, is very true. Now, some examples for the unpredictable or unpredicted toxicities from the past. The large surface-to-mass ratio is apparently thought to underlie the oxidative stress in the lung of ultrafine carbon black. This is one toxicity which was not known from previous medications. This is one of the new toxicities. Then stable fiber-like structure. We just heard about the asbestos-like fibrosis in the lung, for which assessment just heard that work is underway. And there is another unpredictable and unforeseen toxicity. The slow release, delayed clearance by doxil, for example, is responsible for the hand-foot syndrome, quite unknown from previous medications. And the visibility by the immune system, which underlies complement activation. This is something absolutely new with nanoparticles. Traditional medicines, low molecular weight drugs are not recognized by the immune system, and this leads to a toxicity which causes sometimes major problems for the clinical use of some drugs. Now, turning our attention to this complement immune recognition, there is a symptom triad, which we call pseudo-allergy, which inflicts the cardiopulmonary, hematological, and skin system and the skin, and which is the outcome of the immune recognition of nanoparticles. You can see that the cardiopulmonary systems, hypertension, hypotension, arrhythmias, dyspnoea, angioedema, sign of hypoxia, are present almost uniformly in animals and in man. Likewise, the hematological changes, leukopenia, initial leukopenia occurring within minutes, then turning into a rebound leukocytosis, thrombocytopenia, lymphopenia, are hematological toxicity, which is a new phenomenon due to the immune recognition. And the skin reaction, erythema, flushing, rash, all these, again, signs of immune recognition. And these symptoms are shared by a large variety of medicines already in clinics, nanomedicines in the clinics, and also nanoparticles which are under investigation for potential therapy. And these include the liposomes, and interestingly, regardless of the, almost we can say that regardless of the structural features of liposomes, we see the same reaction. Let the liposome be small, unilamellar, pegylated or not, MLV, targeted, the same triad, reaction triad is seen in man and animals. Likewise, solid lipid nanoparticles, micro nanobubbles, polymers, dendrimers, carbon nanotubes, they all cause the same, which led us to the concept that CARPA is actually a stress reaction, very similar to the classical stress. Namely, it is a non-specific, uniform, adaptive response to a large variety of stimuli. And there are many more similarities, but the major difference between classical stress and CARPA is that the axis is different. It is the hypothalamo-pituitary adrenal axis in the case of stress, whereas the complement system, mast cell, cardiovascular system in the case of CARPA. On this basis, I would dare to say that the CARPA, or this immune toxicity, could be perceived as a stress on blood, which is a relatively new concept and obviously requires a lot of feedback. But this is the most recent advance in this territory. And let me just remind you of the medicines which are now in clinical use and show these symptoms. Liposomal drugs, micelle solubilized drugs, antibodies, pegylated enzymes are typically perceived as nanomedicines. Other drugs also cause such reactions, although they are not perceived as nanomedicine. But the bottom line is that it is a very general phenomenon in pharmacotherapy. Now, the significance of blood stress by nanoparticles is listed here. Pseudo-allergic responses to almost all nanomedicines and biologicals represents between 2 to 30% of patients. I mean, it arises, it is observed, which is a very high ratio. A classic allergy, usually the occurrence of symptoms is at most 1%. So it is very frequent. And although it is most cases is mild and transient and controllable, in rare cases it can cause death, which is obviously not tolerable. Standard toxicity and safety tests do not predict the rise or severity of reactions, and I can tell you that for a drug submission, at least 1 to 2 dozens of various toxicity tests need to be performed, and none of those are informative about CARPA. Reactions may surface only in phase 3, phase 4 post-market surveillance, leading to drug withdrawal. This is a major problem for drug developers in the field of nanomedicine. And reactions may contribute to immunogenicity, which is another barrier to the clinical use of nanomedicines. Once nanomedicines become immunogenic, it turns actually into a vaccine, so unlikely usable anymore. For all these reasons, testing for CARPA is very important, and this was also recognized recently by the EMA in their latest reflection paper. They refer to CARPA as a test recommended for the development of liposomal products, and we believe that very soon such recommendations will show up for other nanomedicines, not only liposomes. These are, when the EMA or any agency recommends CARPA tests, and one looks in the literature, what kind of tests, what is this concretely, the only thing which is officially found is this pattern, this list of assays for the hemocompatibility testing of medical device, usually endovascular grafts, shunts, all these medical device listed here. And here you can see complement activation assays listed. Almost all available ELISAs are shown as possible tests for using, but this is for a medical device, not for nanomedicines, which are usually in solution. So there is no, at present, officially approved or validated CARPA test. For this reason, we have been compiling a list of CARPA assays. As you can see here, there are some in vitro tests looking for complement activation by available ELISAs, in vitro using human serum. And most recently, the latest development in this regard is the development of a pan-specific ELISA for measuring complement activation, which enables assessing complement activation in almost all vertebrate or possibly other species. Now, the in vivo porcine and dog test, Dr. Urbanic was talking about it in detail in the previous session. I wouldn't go to the details. We are measuring a lot of parameters at the same time, and this assay is considered, we believe, we suggest as a quantitative assay for nanomedicine-caused reactions. These are some of the changes which we see in pigs. We measure at least a dozen different parameters at the same time and find very close correlation between complement activation in vitro and the reactions caused by these medicines in vivo in pigs. So this is the PAN-C assay, which was introduced 2 months ago by Quidel, and we find very close correlation between complement consumption, here shown on the lower curve, and the drop of blood pressure in the pig injected with zymosan, and similar data are available for rats, showing the utility and the quantitative nature of this assay. Finally, this is my last slide. We developed a decision tree for CARPA prediction. In the first step, we look for complement activation in vitro in normal human sera, in a few human sera. And if there is no reaction, then for safety, we recommend the drug being tested in vivo in the porcine test. If there is reaction in this assay, then it means that there is a likelihood for proneness for CARPA in vivo, so the drug should be changed or the development stopped. Going further on the tree, if in the animal test the animal shows reaction, then either stop the development or apply another administration protocol, for example, slow infusion, or do something about it because there is a likelihood for human reactions. If it is the negative in the animal model, then it should be safe at least for immunogenicity or immunoreactivity. To summarize my messages here, classical toxicity assays may not be applicable to predict possible new toxicities of nanoparticles. New assays protocols are needed, and it appears that for each new nanomedicine, each new nanoparticle, different assays may need to be considered. And CARPA, which is a new nanotoxicity, represents a showcase where new tests and protocols are being developed and introduced to the practice for researchers and drug developers. Thank you very much. This is my acknowledgement for, and if you have questions, then.
No? Yeah. I just wonder if you have seen any reactions in pig that you could classify as false positive. The reason for asking is that I 20 years ago worked with particles that gave very strong reactions in pigs, probably due to their pulmonary intravascular macrophages, and we didn't see any reactions in any other animal models or in humans.
Yes, the porcine CARPA model represents a model. Okay, the porcine CARPA represents a model of the hypersensitive people, not the normal people. So when there is a reaction in the pig, it implies that sometimes, somewhere, out of many, many people, someone will react. So it's impossible to tell whether this is false positive or false negative at the time until a huge number of human data become available and none shows reactivity, although we see the reactivity in the pigs. So the point here is that this is a model of hypersensitive people, not the model of man in general.
Thank you. Just to comment, the related reactions in pigs really do not mimic at all CARPA reactions due to complement activation in pigs.
Okay, thank you. Yes.
Janos, in the last 20 or so years of using Doxil, how many deaths have been absolutely ascribed to this syndrome?
Okay, the box label of Doxil, the first warning tells that potential death. There's no statistics. I am absolutely not aware of it, but if the label already talks about death, it means that sometime, somewhere it happened.
Yeah, I suspect that the actual number of deaths is, if any, is extremely low.
I know statistics about it, about 0.1%.
Yeah. Because with the use of pre-medication and slow infusion, nowadays, I think that the risk of death is extremely low.
Yes, in general, 1% of people who undergo anaphylactic reaction they cannot be salvaged. So, and anaphylaxis is a relatively rare manifestation of allergy. So about all hypersensitivity reactions, 1% leads to anaphylaxis. So 1% of 1% of all hypersensitivity reactions might be lethal. Yes?
You were saying that there are pseudoallergic responses in almost all nanomedicines. I know for liposomes it's quite well described, but for other types of nanomedicines, do you have data as well, or?
Well, to my best knowledge, only liposomes are in clinical use among nanomedicines. Obviously, for anti-cancer drugs, which are solubilized by chemophore, for example, or other emulsifiers, these carriers are nanoparticles, and there are a lot of reactions. I can tell you that those patients cannot be treated without pre-treatment against the reaction. So it's very frequent.
So I think there are a couple of other products besides liposomes.
Okay,
and I was interested to know if you, because you could postulate that probably for other...
Yes, because I am not very much aware of nano products in the clinics. What do you think of? Which one?
Iron oxide, nanoparticles. Micellar products.
Okay, well, iron oxide, very good example, because about half a year ago, the EMA prohibited the treatment of patients with iron in the absence of equipment for immediate help of the reactions, because it occurs there. So much so that a case recently in France triggered a huge a committee came together and discussing the iron reactions.
Okay, well, I think we could debate longer.
Okay, well, I have...
There are pseudoallergic reactions against almost every drug on the market, including low molecular water soluble molecules, as you listed.
Well, they are referred to...
When you ask about pseudoallergic reactions,
yeah.
you should expect to have that for almost every compound you try.
Well, when you talk about infusion reactions, hypersensitivity reactions, it has a lot of synonyms. Then they are always the same phenomenon.
No, and not always the same.
Well, I'm talking about the rapid phenomenon, the one which arises within minutes.
But I am speaking about the mild pseudoallergic reactions you mentioned.
Yes? Okay, well, it has a broad scale, mild to severe. Okay, all right, last.
Hello, do you have any information on which pathway is being activated, and is it nanomaterial specific?
Yes, we know that all complement pathways can be involved in triggering this reaction, even the lectin, which is new, and the next speaker, Professor Mogimi, will probably talk about it.
Okay, thank you.
All right. Sorry. So this will be the last.
Janos, very brief questions. Except porcine models, what else could be used? Let's say rabbits or sheep, dogs?
Essentially, most animals can be used, except their sensitivity differs tremendously. Pigs are by far the most sensitive. Dogs also react, but with less sensitivity, and not every dog reacts to the same trigger drug. Rats, there is a poster here about rats. Rats also show symptoms, but at a dose about 1,000 times higher than the pigs. So this is a general phenomenon. It is present in most animals, but the manifestations differ, okay, and the sensitivity.
Okay, and final question. Do you find in your career any nanoparticle, either organic or inorganic, which do not activate complement?
Absolutely, yes. Yes. We see particles which do not activate complement and doesn't cause any reaction in pigs. And actually some of those are developed in our consortia. So yes, there are.
Okay, thanks.
Moin, my friend. Professor Moin Mogimi from Copenhagen University is going to talk about